Hello everyone, welcome to this channel, and today I am going to give you briefing about the Abo blood grouping. So in the earlier centuries the blood transfusion is done, and in some cases there is occurrence of transfusion reactions, but back then we don't know anything about the antigen or antibodies. It was in the year 1901 when the Austrian scientist Karl Steiner, he actually find or observe that there is presence of antigens in the surface of the RBCs, which are actually responsible for the agglutination or clumping of the RBCs, which causes transfusion reactions. So he classified two antigens, that is antigen A and antigen B, and he found that these are present on the surface of the RBCs. He also found that there is the presence of corresponding antibodies, and he named them as A or alpha antibody and B or beta antibody. And one thing is there, the same antigen and its corresponding antibody should not be present, and it is not present actually because it will cause transfusion reactions. So for this work he is honored with Nobel Prize in the year 1930. So based upon the presence or absence of these antigens, blood grouping is done, and there are four blood groups, that is blood group A, blood group B, blood group AB and blood group O. In case of blood group A, there is the presence of A antigen in the surface of the RBCs and presence of beta antibody in the serum. In case of B blood group, there is B antigen in the surface of the RBCs and alpha antibody in the serum. In case of AB blood group, there is presence of both A and B antigen in the surface of the RBCs but no antibodies in the serum. And lastly, in case of O group, there is complete absence of any antigen on the surface of the, that is absence of A or B antigen on the surface of the RBCs and presence of both alpha and beta antibody in the serum. So this is the Landsteiner law, that is when an antigen is present in the surface of the RBCs, the corresponding antibody should not be present in the serum. So blood grouping or knowing the blood group is necessary for basically the blood transfusion. So for that, blood typing is done or blood blanching is done, and it is based on the principle of agglutination, that is reaction between antigen and antibody which causes clumping or agglutination of RBCs. For that, the blood sample of the individual to be tested is taken, and some of the drop of it is mixed with saline to make a suspension, and two other anti-sera test samples are taken. One consists of anti-A antibody and the other one consists of anti-B antibody. So what we will do, we will take a slide and we will take one sample, one drop of anti-A in one portion, that is in one side, and anti-B, drop of anti-B in the other side, and we will mix the RBC suspension with that. We will mount that for 2 minutes, and after that we will check something like if there is presence of any clumps or masses, if it is clear, these things. So if we find that there is clumping when we mix the anti-A with the, when we mix the blood group with anti-A, so it is blood group A, because there is the presence of corresponding antibody or isoagglutinin alpha which causes the agglutination reactions. And if we find that the clumping occurs with anti-B, then it is blood group B, because it consists of B antigen which actually react with the beta antibody. And if we found that both anti-A and anti-B mixes results in clamping, it is AB, because it consists of both the antigen, that is both A and B antigen. And if we found that there is no amount of clamping and it is clear by both naked eye, or if we take the microscope it is clear, then we will say that it is blood group O, because there is no antigen in the RBC surface. That's why no reactions occurs with both anti-A and anti-B. Now this is very necessary before blood typing or before the blood transfusion, because same blood should be given to a recipient, that is the one whose blood is taken is known as the donor, and who will take is the receiver. And one thing is considered every time, that the antigen of the donor and antibody of the recipient should be considered. So this is a brief idea regarding ABO blood grouping and why it is necessary. And here I am ending my today's video, and if you guys like my videos, please subscribe to the channel. Thanks for watching.